spotted a small correction with my department. I was in the Department of Electrical and Electronic Engineering until Tuesday last week. And I am now the head of the brand new Department of Biomedical Engineering, which started on Wednesday. Okay, so this is what I'm presenting now is definitely a team effort. Okay, I suppose it's the expectation that professors will present other people's slides. But I did make some of these slides myself. But I want to acknowledge, and you can see there across the bottom, that we have the University of Melbourne, the Florey Institute of Neurosciences and Mental Health, and the Royal Melbourne Hospital. These are the three major partners in this research. But there are other research organizations involved as well. And even within the University of Melbourne, it's electrical engineering, mechanical engineering, biomedical engineering now, Department of Medicine. And then there's also Smart Stent and Synchron, which are two companies based in Australia and the United States, respectively, that are now involved in this as well. So, if I can get that to work. Okay, there we go. So paralysis is a very debilitating condition, and we can see there are some examples of these. Pardon the slides are a little bit stretched out, but I think it'll be all right. Also, I'll make an excuse straight away. They haven't been able to test my videos yet, so hopefully they're going to work okay. All right, so 60,000 Australians, 6 million Americans. You could probably scale that to New Zealand as well. 50 million globally by 2025 is the expectation of people that will suffer from paralysis. Spinal cord injury is one of the major causes of paralysis. And it's not just that there are so many cases and so many new cases, but also most of the injuries are occurring early in life. And so it affects people for a large part of their lives. And also, it, in most cases, results in an early death. Probably most of you are familiar with Christopher Reeve, who's one of the most famous examples of a spinal cord injury when he came off a horse and injured himself, his neck. And he died not too long after, just a number of years after. And there is no cure at the moment for spinal cord injury, although there is a lot of work going on in using stem cell therapy and others to help repair the spinal cord. Another area that has advanced an enormous amount over the past decade or so has been assistive devices for people who have spinal cord injury. So here is an example now that comes from Ethor Bender's TED Talk in 2011, showing this sort of early version of an exoskeleton for walking. You can see there, obviously, there's a number of things that still need to be developed, but they have developed a fair bit since then. One is that the device is, well, she needs crutches to be able to maintain her balance. It's quite slow and cumbersome. But probably the main thing that doesn't really come out in that presentation is that exoskeleton is being controlled by the man that's following her. And apart from some internal control that is sort of maintained by sort of balancing and sort of the automatic movement of the legs when it moves forward, she doesn't really have any thought control over those legs. So brain-computer interfaces, or brain-machine interfaces, are developing ways of being able to acquire signals from the brain, or if possible, from peripheral nerves or the spinal column, decode those signals, and then use them as commands for these external devices. And we can see there are a few possibilities. Loss of limb is a possibility, wheelchair control, these leg exoskeletons, and also just computer screens, computer mouse. There's a number of technologies that can be used for recording brain signals. The least invasive is EEG, and probably most of you are familiar with EEG. Many of you have probably had an EEG recorded, I'm sure. This is a rather extreme example I'm showing here. But you can see here you get this a signal that has some it's generated by neurons. It's not the neural signal, but it's more like an artifact of the neural signal. And that signal is only at low frequency and very low resolution when you record from outside the head. So we can see in this case that for electrodes outside, the skull attenuates the signal a lot and also disperses the signal in space. So the best sort of resolution you can get is in the centimeters, which is not really good for fine control of brain-computer interfaces. I always forget to warn people before I turn this on, but this is not too bad. A picture of somebody's brain with electrodes placed on the surface of the brain. So these are now subdural electrodes. You can also do epidural, nearly as good. But with invasive open brain surgery, you can place electrodes directly on the surface of the brain. So that gets around this issue of attenuation. You can record higher frequency signals, and you can record at much better spatial resolution because the electrodes are touching the brain, touching the cortex from which you're recording. So you can get down into the sub-centimeter range with the sort of best electrodes that have been developed so far. However, they're very invasive, and there's around about a 1 in 4 chance that there'll be a surgical complication from the neurosurgery. Often that is epilepsy. Now, rather than recording the sort of the fields that are generated by neurons, it is also possible, using arrays like this one, which is a Utah array, to actually push electrodes directly into the brain and record from individual neurons or clusters of neurons, or also what's called the local field potential. So these have by far given the best outcomes for brain-computer interfaces so far. However, they are even more invasive than those subdural electrodes because they breach the blood-brain barrier. You're just physically pushing these spiky things into the brain. And also, over time, the electrodes are rejected by the brain. There's a lot of glial tissue growth and eventually some electrode failure. So this video here gives an example of a person who has these devices. So maybe you can see there's a couple of connectors plugged into the top of his head, and you can see he's got this pretty amazing control over that simulation of a robot arm. I think it's a simulation. No, actually in this case it is a real robot arm. So that's quite astonishing. You can see the level of dexterity he has and the ability to very quickly target the square or point at it. What is not clear from this video, because it actually comes without sound, is probably the most remarkable thing is that he is chatting with his girlfriend about something completely different while he's doing this task. That is the holy grail of what we're after with brain-computer interfaces. Can that arm or that thing that we're controlling become an extension of our own body so that we can do something else at the same time and not just have to think, move the arm, move the arm. Just like I'm doing at the moment. I'm pacing back and forth while I'm talking to you, and I don't even think about it. The issue with these devices is not the technology. So DARPA put a lot of funding and research and development into developing these arms that have many degrees of freedom. They look and act very much like human arms. They still break very easily at the moment, so there's still a bit of work to go on that. But something to control is definitely already there. The problem here is seen by that red line. The red line is those penetrating electrodes. And you can see there that there's a lot of information that can be obtained from the brain, and that improves as algorithms improve, as the electrodes sort of settle down over a period of a couple of months But then, within about a year or so, most of those electrodes have failed. So in some cases, people have been able to continue to use this beyond a year, but they're really only able to gather signal from maybe 1, 2, or 3 of those 100 electrodes. On the other hand, the blue is the surface electrodes, those still invasive ones. They actually take a few months to settle down and to get a steady signal. But you can see here that even though they last a very long time, the amount of information you can get from them is fairly low. So we want to aim for up here. So what our aim is in this project is, first of all, to increase safety. So reduce the invasiveness of placing electrodes in the brain. In particular, no open brain surgery. We want to maximize the performance. So to do that, we need to get the electrodes as close as possible to the neural tissue. We want to ensure functionality. So we want to be able to put the electrodes where we want to put them. In particular, if we want to control a prosthesis, we need it to go near the motor cortex. And there are other areas as well. The posterior parietal cortex and supplementary motor cortices, but probably M1 is the initial target, primary motor cortex. And chronic reliability. So this means not breaching the blood-brain barrier, something that can last for the rest of a person's life. So those are tough. So nothing developed so far meets all those criteria. So what a very bright young spark a PhD student found was that he was working, he's a neurologist, and he was working in this new technology called the stent retriever. And that's a technology where you can put a stent into a blood vessel of a stroke patient, expand it into the blood clot, and then withdraw the clot from the brain. So interventional neurology. Knowing you could put a stent in there, he thought, well, can we actually functionalize a stent and use it to record? And the first thing he looked at was to see, are there patent large blood vessels close to the areas that we would want to record from? And sure enough, there are. So we can see here, the motor cortex here is shown in red, and you can see that there's this large superior sagittal sinus vein that passes up through the middle of the brain, and then this central sulcal vein which passes down over the motor cortex. So the idea was, can we functionalize a stent, put some electrodes on it, put it into these blood vessels and record from them? And yes, so that's what I'm going to be presenting here, is this device. So we published this in April last year in Nature Biotechnology. Well, I'm not sure how the sound's going to go with this, but I'll just get a good mate of mine to explain it to you.
Hi everyone.
Yeah, I'll turn the volume up and. Oops.
Hi everyone, this is President Barack Obama. The U.S. and Australia are funding research into a device that could someday allow people to control their prosthetics with their minds, without invasive brain surgery. When implanted into a blood vessel, this network of tiny electrodes can record your brain signals, technology that has the potential to transform the lives of our wounded warriors and others with disabilities. It sounds like something from the future, but it's just another example of how investing in research can move us all forward.
Now I promise you that wasn't lip synced, even though it looked like that. There's something funny there with the video and the audio, but. Okay, so for the rest of this talk, I'll just take you through some of the properties of this device. So we've called it the Stentrode and trademarked that. So a self-expanding electrode array. So the key is it has to be very, very small, very thin, to be able to be delivered to the right location through blood vessels. But then it needs to expand out to be able to fill essentially the perimeter of the blood vessel, the outer wall of the blood vessel. So here you can see an example of a catheter that has the Stentrode through it. As you push the Stentrode through, it's attached to the Stentrode, to the stent at the front. You can see that it's made of nitinol, so it has this sort of it has a shape memory, and it will automatically expand and deploy into the blood vessel after it comes out of the catheter. Why aren't I advancing? Okay, so we're looking at this as an option for patients with paralysis, and the idea is that when we test it with patients, we will first of all non-invasively image the areas that we need to put the stent as much as possible. So one example is to place them into an MRI machine, do functional magnetic resonance imaging, and get them to imagine moving their legs, for instance, and then look at which regions of the brain are being activated. And that will give us a clue as to where we want the Stentrode to go. This is still quite an important topic of research for us. We don't want it to slow down getting into our first in-human trial, but we're doing a lot of work to see how well we can localize these regions in paralyzed patients, especially those who've been paralyzed for a long time, in which case their regions of the brain might not be so active when they're thinking about movement anymore. Then the hospital procedure is actually just a day procedure. It's very straightforward. It's being done a lot. Well, it was originally developed for heart surgery, of course, the stents, but it's often being used for stroke surgery now. And it's within the sheep, it is down to just a couple of hours all up to get one of these devices deployed. Access through a vein, so essentially into the jugular vein. It's quite easy to get in there, and we're certainly not opening the skull. So the way it works is to actually feed up a wire initially. There's a wire that has a little bend on the end of it so you can steer it, and it's called interventional radiologist actually passes up that wire and guides it through using angiography to the location it wants to go, and then a series of catheters are then fed over that wire of increasing diameter. So starting off with a very small catheter, and then a thicker, and then a thicker, until we get to the it's called the 6 French catheter is the size that we want, and then you withdraw all the catheters from the middle, and then you can deploy the stent through that. And I've already shown you this the self-expanding stent. So here's if I can get this to work. Is my pointer gone? A little video that we made to sort of illustrate this. So part of this video was shown by Barack Obama. So you can see it there. You can actually also see it in the video there how the electrodes have been sort of interconnected by these wires. There's a wire that terminates at each one of those electrodes and eventually becomes a bundle. At the moment, there's a requirement for the wire to actually exit the neck, and then we have connectors to connect onto that wire. Now, the electrodes are close to the brain regions we want to record from. So the idea then is, say, think about moving your legs. We interact with them and try then to build a machine learning system that will interpret the signals that we're recording from those electrodes. So we need to get as many electrodes in as possible while still maintaining a safe device. There's a lot of limitations in the number of electrodes that we can place, mainly because of the wire that needs to come out. We can't allow that wire to be too thick. After we've acquired and transmitted the brain signals, we can decode them and then translate that into these many devices that are currently available for use by people with paralysis or loss of limb. So this figure over here shows the configuration that we're likely to use for the first-in-human trial next year. So we want a wireless communication with the device to minimize the likelihood of infection, but there will still be a wire that comes down into the chest area. I think it would take us far too long to build a wireless system just based in the stent. We would run out of money too quickly. Okay, so in terms of developing the procedure, what you can see here is angiography. So what you can actually pump, I don't do this, but forcefully push a dye up into, actually you're pushing the blood backwards in order to do this. And that allows a very high contrast image to be taken. Take a photograph of that image, and then in real time, you can watch the wire making its way through that topography that was just mapped. You can also see there the telescopic system. So this here shows that bent wire at the end that is used to navigate, and then all the different catheters that have been placed over the top of it. This is the sheep brain. So the reason that we've used sheep is because they actually have heads of a sufficient size. They've got blood vessels they're still smaller than human blood vessels, but much larger than blood vessels of say, dogs or monkeys. Another advantage is that the motor cortex of the sheep actually runs in parallel with the central vein. In humans, it runs parallel with that branching vein, but the sheep have got a motor cortex that runs front to back. So that's very handy. And the diameter of the superior sagittal sinus is about 2 millimeters. Remember, in humans, it ranges between 4 to 6 millimeters, so quite a bit bigger. What do you think might be a disadvantage of using sheep?
You can't tell them to move their legs.
Yes, you can't tell them to do anything. Yeah, so other animals can be trained to do things. Sheep. We've tried. We have tried. We actually have had some success. We're trying to publish that, but it's frustrating. You try to. We've managed to train a small number of sheep to do a feeding task. Actually, I'll show you it in a moment. And so we thought, well, we should publish this. And they say, the main comment we get back, you haven't done enough sheep. But none of us can spare the time to do more sheep just to show that they can learn something. All right, so 2-hour procedure, as I mentioned. This is now looking across. You can see the Stentrode will be deployed along this region. Those blue lines show some of the sort of the turning points and the end point of where we want to go. This turning point here is quite a challenge for the surgeons to get around. This last one actually shows, we can see here there's a bit of a guide, but you can actually see the wire coming through here and the stent there with the electrodes on it. So does it work? Well, we used a number of different measures to see. One of the measures was to day 0. So at the time of deployment, we would hook up the stent while the sheep was being brought out of anesthesia, and we're able to sort of see that progression of the change of the signal over time. And so here you can see a small bit of that waveform. It looks very much like those intracranial EEG signals that we saw before. The second part of this graph, so this shows burst suppression ratio, which is the measure of the EEG variability on that day 0. After 1 month, the sheep were anaesthetized again, and the same measure was made. And you can actually see here, there's a much stronger, more reliable signal. So we were quite excited about this, that first of all, we can record that the device was patent out to a month, but that also the signal actually improved over that month. We also, because the somatosensory cortex is close enough to the stent, we also would stimulate the forelimb of the sheep and record the evoked potentials in the brain. And here you can see an example. So this is there's a stimulation artifact that we've picked up, and then you can see here at the different electrodes of the stent different aspects of that recorded neural signal. In this column here, we can see that same experiment being performed on day 1 through to day 28. So day 28 looks a bit strange, but you can see that as it goes, the amplitude is gradually sort of increasing. This here is largely because the early versions of the devices, the wire that connected the stent to the outside world broke very easily. The main reason for that is that it passed down near the jaw of a sheep, and sheep chew all the time, and eventually that would break the wire. Chewing artifact was another thing we picked up a lot. Okay, so this is our attempt at training sheep. So we had an automated feeder where we would provide a touch sensor, so the sheep would touch that when they saw a light, and on doing that they would then get another light showing them which side the feed was going to come, and then they would move their head either left or right to collect the feed. And so that was an evoked movement in the sheep. But it's really simple. See, it doesn't prove a lot, but you can see that we tend to get those standard sort of gamma activity that is normally used for brain-computer interfaces. So that's a spectrogram that's shown on the right. To compare with other devices, we looked at some objective measures. One of them is the useful bandwidth. So what we can see on this graph is an epidural array. So this is an array that's placed under the skull, but on top of the dura. The middle one is the stentrode. The bottom one is a subdural array, which is the gold standard for electrocorticogram, or the EEG from inside under the skull. And that's placed under the dura, directly on the surface of the brain. So basically what we did was to look at what frequency could we go out to before we got to the signal being lost within the noise floor. And you can see here that what we call that bandwidth. The subdural array has the best bandwidth, as we would expect, but the stentrode is about the same as an epidural array, and they're both at 200 hertz anyway, which is about what we need. Most of the signal is under 200 hertz. We can also see power measurements that are comparable between these different devices. So we've shown that it is comparable to the existing more invasive approaches. Actually, this figure shows an X-ray with those other the subdural electrode arrays in place. So they're much bigger electrodes, and the stentrode is just these tiny, you can barely see the electrodes in the X-ray or CT scan. Another area that we're interested in pursuing in the future, but we just tried it out in a couple of sheep, was electrical stimulation. Can we stimulate with this device in the blood vessel? Because that's going to have a lot of therapeutic possibilities in the future. And here's an example of a sheep where we could stimulate a part of the motor cortex and its lip would twitch. But in that preliminary study, that was sort of about all that we could get. There was some further work that was done looking across the different electrodes and comparing to different locations but not as much as really understood about the neuroanatomy of the sheep brain. So we were sort of playing around in the dark. We're currently, I won't talk about it more in this study, but we're currently doing some work where we're actually placing the electrodes into the visual cortex area. We can measure responses to light flashes, and we can also stimulate within that region and then use subdural arrays to record the neural responses to that stimulation. So that's just started. Safety. This was the most critical thing. So we needed to show that the device is safe, most important, and that it could record a useful signal. So I've shown you the useful signal already. What we see here is we took sheep out all the way out to 20 weeks plus, and we can see here that that maximum bandwidth is staying pretty steady across the duration. And so that shows the signal stable. This shows that it's safe. So in vivo lumen diameter is basically what is the diameter of the blood vessel after this device has been placed. We do not want it to occlude the blood vessel. But we can see here that compared to the standard diameter, which remember I said was about 2 millimeters, we can see that it stays pretty steady, maybe even increasing a little bit, which would be the action of the stent, sort of pushing it out. What we actually found is that the device becomes incorporated within the vessel wall. And this has been known for a long time about stents, is that the vessel wall sort of endothelializes. It covers over the electrodes. So we tested this by taking a couple of brains down to the Australian synchrotron, doing some micro CT, and you can actually see here so this is how we sort of measured the diameter of the blood vessel, but you can see that the electrodes, which is, you can see the reflections of the struts of the stent and of an electrode here, are actually outside that diameter. And we think this is the reason that we actually get a signal that improves over time, because our electrodes are being insulated. They no longer have blood passing directly over them. So that vessel incorporation usually took about 2 weeks or less, and even out to 190 days, we saw no vessel occlusion occurring. So this was tested by taking the sheep out to increasing amounts of time before checking their blood vessels. So it looks like it's safe. So where to from now? Well, we've done quite a bit of preclinical work that has led us to know the sort of parameters that we need for the device and the initial studies of safety. However, this device is certainly not suitable for human use. You know, we just make it on a bench in a small room in the Royal Melbourne Hospital. So we're now developing a human quality version of the device, which we will then test in long-term preclinical work, continuing in sheep. Then we'll move into the clinical. So we need to develop the sort of brain-computer interface algorithms, control algorithms that we want to use, and also go through a process of patient selection and training. There is no shortage of volunteers, actually. We have plenty of people who are volunteering to try the device, but we need to make sure that we pick the right ones. The importance of that has been highlighted, you know, in the first bionic ear testing and the first bionic eye testing, that those initial patients are really crucial. And then early 2018, we'll be moving into our first in-human trial. So primarily that's for safety, testing, but also we want to demonstrate the effectiveness of the device. So we need to take these brain-computer interface control algorithms that we've created and adapt them very rapidly to the human, trying to minimize the amount of training time that we need with the humans to be able to learn these, to use these. All right, so I think I've pretty much covered most of these, and probably most of you know what's going to be required in the preclinical stage, but we need to ensure good manufacturing practice. We have to document this for the Food and Drug Administration, including the workflow and everything. So it works out to documentation all about that thick. We need to show safety, so biocompatibility, well, that's largely done because nitinol is known to be biocompatible. The platinum that we use for electrodes is biocompatible. We use platinum wires, so it's all been shown already. We need to test for accelerated aging. So we actually are going to bend our devices many, many times and put them through heat tests and so on to show the glue won't break down and so on. MRI compatibility, we've talked about this a lot. Does having this device mean that somebody's never going to be able to have an MRI again? Well, we need to find out. So maybe low field MRI will still be possible, certainly not high field MRI. Corrosion, yep, so that's part of the accelerated aging, testing that. How do we sterilize the device in a way that's not going to destroy it? And the stent delivery and implantation. So there's a lot of training that the radiologists will need to do to ensure that they can get it in quickly and safely and into the right spot. Ideally, we'd like them to be able to turn the device to the right angle, but that seems impossible. And then device efficacy. So what's the recording capability of this final human quality device that we manufacture? The longevity and the stability of the recordings, and what sort of information content can we get from it? The patient selection we need to consider. The injury level. So generally, a patient has more to gain, less to lose the more injury they have, but the more difficult it will be to work with a person who has a very high level of injury, or a very high level of spinal cord injury, for instance. Time from injury is important. If it's too long, the brain may have reorganized and not be as easy to obtain a good signal from. And then also time commitments. We need these people to be coming into the lab, hopefully multiple times a week, so that we can develop the tools, the signal processing. We need to decide what tasks, what is the best task that we're going to ask the patients to do. Perhaps the easiest would be just to control a mouse on a computer screen. But is that really what a patient wants to do? We need to consider actually providing some benefit to the patient as well. What algorithm should we use? Is it best to keep it as simple as possible so that it's easy to adapt, or do we want to go with more sophisticated, maybe Kalman filtering or convolutional neural networks or something where it may be difficult to get enough training data? So as I said, the human trial is mainly to validate the Stentrode, to show stability of the device versus the signal that we record. The FDA is going to be primarily interested in the safety, but there needs to be efficacy to get approval. We'll examine how the patients learn to use the device, not just how we can learn from the patients, but how the patients adapt as well. And also, maybe we can measure some neural changes that occur as the device is used. We know that occurs with bionics. With bionic ear stimulation, there is reorganization of the brain that learns the new signal. Finally, we want to eventually take this to a clinically relevant device. Having a wire that comes out is not the best, because we're going to have a wire coming out of the jugular vein for the rest of the person's life. That's not particularly good. So we are now working on how we can wirelessly transmit data. So we need about 100 megabytes per second. If we're going to have 1,000 electrodes at 20 kilosamples per second and 4 bytes per sample. That's pretty high res. That's probably more than we need. So that's probably the upper limit of what we're after. Hermetic encapsulation and feed-throughs. Well, this is something we have to solve anyway for the first in-human device. And but fortunately, there's now a number of manufacturers around the world who do this. So we're tapping into that. And then finally, the stable recordings and stable algorithms. I've mentioned those a few times. Finally, the future applications. I talked about stimulation. I'm actually just as excited about the stimulation possibilities of this device as the recording. For epilepsy, we would want to do both. So we've been working in epilepsy now for epilepsy research for about 12 years now, and in particular, looking at how we can predict and then suppress epileptic seizures. So one of the hindrances of this is the invasiveness of placing the electrodes where we need to in order to measure and then deliver the therapeutic stimulation. We don't want to make the epilepsy worse by opening up the skull. So perhaps this is going to be a way to do that. Other deep brain stimulation possibilities are potentially possibilities. And so these are known to benefit from deep brain stimulation. Less well known and still under development, post-traumatic stress disorder, depression, obsessive compulsive disorder, and some others could well be treated by this, what's now being called electroceuticals. So we're writing grants on this stuff as I speak. So if you get a grant to review, be nice. Okay, that's it. So I'd like to acknowledge these partners. So Tom Oxley is the one who came up with the original idea. He subsequently went on and did his PhD, held on to it until we got the Nature publication, and then his PhD passed with flying colors. The other key ones to point out here, I think, are Nick Opie. So Tom and Nick together have formed these companies, Synchron and SmartStent. And SmartStent's not shown there. And then also Professor Terry O'Brien, who's a neurologist at the Royal Melbourne Hospital and Department of Medicine. And especially my postdoc, Sam John, who's done a lot of work, mainly around the signal processing aspects. But thank you very much for your attention.
Fascinating talk. Thank you very much. So, questions? I think.
Oh, sorry. Yeah. I have two questions, and that's a very interesting talk, by the way. So my first question is, how did you decide on the number and the spatial arrangements of the electrodes? Is it basically limited by the diameter of the wire, I guess? The second question is, earlier in your talk, you showed that most of the electrodes basically fail after 1 year. How do you think this device will perform for that period compared to existing electrodes?
Yeah, sure. So the first question was about the designs, really. So we can only really place the electrodes where the struts sort of meet each other in the device, and so that sort of limited where to put the electrodes. They could be put elsewhere, but then there wouldn't be as much for them to be glued to. What really limited the number of electrodes, as you said in your question, was the wires coming out. So, you know, as you add more electrodes, you add more wires to your bundle, and it gets thicker. It becomes harder to push it around the bends and so on.
You want 1,000, right?
Yeah, 1,000. Well, that's not 1,000 all in one stent. So eventually, you know, we would be looking to maybe deploy several stents. I mean, 1,000 is dreaming. No, that'd be great, but yeah. When it's wireless, then, I mean, as long as we can power it and get the data transmission rates high enough, then because there aren't those wires, it becomes possible to put a lot more electrodes onto the stent. The second question was about the wire breakages. That problem has been solved now. So it was actually, the main point was this, where it curled around a lot. That doesn't exist in humans as much, so that's not as much a concern. But the other is we found better ways of twisting the wires together. We had a lot of trouble actually with the connector on the external side and being able to tap into the wires, but we've solved that with an improved connector as well. It's funny when you launch into these projects, which are, let's see, to develop the device, it was over $1 million. And we thought our challenge is going to be making the electrode stick on the device, getting it in, and recording something useful. We spent an inordinate amount of time fixing the connector at the other end. It was such a problem.
Another question back there?
Thank you for the great talk. And my first question is, what's the maximum sampling frequency that you're working with at the moment?
So we're working at 1,000 hertz sampling frequency, where we know we can't really record a signal much over 200 hertz anyway, although we do want to see what we can get up to about 500 hertz, but we think a kilohertz is probably enough.
And are the events, are the events different in terms of their profile for different tasks? It's like signal processing question. Yes. Well, yes, they are. So activating, say, different fingers or different muscles of the leg is different locations within the cortex, and so there's a triangulation that we can do there, or closer electrodes are going to record a stronger signal for one type of activation than another. But also, at different stages of the movement, like the planning and the execution and so on, there's activation of the different frequency bands of a signal. And so we can look at the beta, we get a beta deactivation and a gamma activation. And that's the main signal that we're sort of focusing on at the moment. And how do you estimate the origin of those events?
Basically by which electrode is recording the highest amplitude of those events, with the assumption that the closest electrode is going to record the closest electrode to the origin is going to have the strongest signal.
But again, it brings up another question right after this, like in terms of stretching a hand, how far or how intense you're going to stretch the hand, and how are you going to compare those events to this stretch?
Extremely good questions. Yeah. So we actually believe that you can learn a lot more about those degrees of activation by looking at the interactions between different brain areas, and so that's what we're working on now. But it's largely an unsolved problem.
And how long does surgery take?
That surgery, we've got it down to 2 hours in a sheep. Yeah.
Thank you.
You're welcome. Yeah.
David, one of your goals is to have electrodes which don't degrade over time. My understanding with Parkinson's disease is that certainly for a large number of patients, they do start suffering degradation and effect. There's an argument about whether it's disease progression.
Yeah.
But I understand electrodes are usually held responsible. What are your basic mechanisms for why you think your electrodes are going to do better in that type of application?
Well, I think one is because the electrodes are not actually in contact with the neural tissue, that there'll be a much reduced reaction to the presence of the electrodes. So we anticipate no glial growth, so no inflammation or changes in the structures of the brain around that area that just results from having a hard electrode surface in contact with the soft brain tissue. But I think it's probably limited, the amount that the Stentrode will make a difference in terms of that efficacy over time, because I think actually for Parkinson's disease, my understanding is that there is a continuing reduction in the dopamine, or in the dopaminergic neurons, and so that there is a degrees progression there that is being that needs to be dealt with. So I think that one of the major advantages of the device is just that it doesn't require electrodes to be pushed through the brain to get to the regions that you're targeting. If we can go through the blood vessels instead, I think that's the major advantage.
Yes, we can have one last question because it's a lecture that's starting in 10 minutes. Right.
I'm happy to talk to people outside.
Are there other sorts of devices that involve chronic implantation of devices that are crossing the venous wall? Like the wire on this is going to have to go from inside the vein to outside the vein, and that's going to have to last the life of the patient.
Yeah.
Have there been other devices that?
Oh yeah, the pacemaker. So heart pacemakers.
They go inside the vein.
Well, they go inside arteries.
Or inside the artery.
Usually, yeah, and then they have to come out to the battery pack, which is outside the arterial system. So I think it has been shown to be patent for a very long time, but it's not ideal.
Obviously, you can do something wireless.
But also generally, if something does go wrong, a pacemaker is not. I mean, it's hard, but it's not too hard to take out. I mean, you can, go the old way with open heart surgery and take it out, but these stents we are not going to be able to pull out. Yeah, and it'd be very difficult to get in there to remove them. So it's important.
Okay, so we'll close to the formal proceedings, but I think David will be around so we can you can discuss more with him outside. And you probably have a small window tomorrow afternoon after a PhD oral, so if people do want to meet with David later, maybe there's a small opportunity tomorrow as well. All right, thanks everyone.